This video is about action potentials. It corresponds to section 41.3 of the Applied Analog Electronics textbook. Now, action potentials are something specific to individual cells. So let's take a look at what we've got. We've got our lipid bilayer. This lipid bilayer, the cell membrane, doesn't let ions through. So it is basically an insulator. We have got on one side the cytosol, that's inside the cell, and on the other side we have whatever the external environment of the cell is. Now, the action potentials are something that doesn't occur in every cell, but it occurs in active cells, that is to say neurons and muscle cells, and a few other types perhaps also. But we're going to be mainly interested in one type of cell here, and that's the cardiac muscle cells. The action potentials that we get in nerve cells and in muscle cells are similar in type, but not identical in detail. So we're just going to look at the cardiac muscle cells because that's what we're interested in. That's where our signal comes from in the EKGs. The skeletal muscle cells are quite similar, and so we get similar sorts of activity from them. There are differences in the way they're triggered, and so some small differences in the way the action potentials are generated, but the basics are the same. What this lipid bilayer basically results in is a capacitor, because we've got the cytosol here, which is a salty solution, and the external media here, which is also a salty solution. So we've got conductors on both sides of an insulator, basically a capacitor. And the way this capacitor gets charged and discharged is by the movement of ions. And we have two different types of movement. One is from ion pumps. Ion pumps can move a charged ion, sodium for instance, potassium, calcium. Those are the ones that are most important for action potentials, sodium, potassium, and calcium. And the ion pumps can push them against the concentration gradient. So it takes energy to push something against the gradient, and the ion pumps use up energy, generally in the form of ATP, in order to establish a charge difference and a concentration difference for each of these ions. Now, in the sort of the resting state for the cardiac cells, the sodium ions are pumped out of the cell. The calcium ions are pumped out of the cell, but the potassium ions are pumped into the cell. So we've got an excess of sodium and calcium outside the cell, an excess of potassium inside the cell. And this ion concentration gradients are enough to get about a 90 millivolt difference across the cell membrane. So there's about 90 millivolts more on the outside of the cell than the inside of the cell. Okay, ion pumps set up the baseline state, and this baseline state is referred to the cell being polarized because it's got this 90 millivolt charge across the membrane. We have another type of way to move the ions, though, and that's what's called an ion channel. Now, the ion channel doesn't take energy. What it does is when it's open, it allows ions to flow from one side of the membrane to the other, and when it's closed, it doesn't allow it. So the default state for this membrane is that the ion channels are closed, and the ion pumps are working to establish the concentration gradient. Then something happens and the triggering event is different for different types of cells. But basically, there's something that happens that opens up the sodium channels. And when that happens, the sodium now can rush back in. And that causes changes in the balance of the charge. Notice that this will cause positive charge to move into the cell, discharging the capacitor. And these sodium channels open very quickly and allow a lot of sodium through, so that the discharge here happens fairly fast. At the same time, or slightly after the sodium channels opens, the potassium channels open, and potassium starts leaving the cell. Also, calcium channels open, and calcium enters the cell. Now, each of these channel openings and changes in concentration of these ions has different effects and has different timings. The calcium channels are very slow acting. So they take, basically the calcium pulse comes in and it stays there for a long time. The sodium pulse lasts a few milliseconds, less than a millisecond maybe. The calcium channel, on the other hand, can result in a calcium pulse that lasts hundreds of milliseconds. And it's this calcium entering the cell, the increased calcium concentration in the cytosol, that causes the muscle to contract. So this is really the key thing for causing contraction, which is the main output of the muscle cell. Once this has happened, however, the muscle cell can't stay contracted forever, and the channels close again, and the ion pumps start getting things back to the rest position. So this thing is called depolarization, when you have the channels open and everything basically discharges, and the pumps, when they're running, repolarize the cell. The depolarization is very fast, on the order of a millisecond or so. The repolarization is quite slow, and it may take half a second, sometimes even longer, to repolarize the cell. So the combination of these two things is what gives you a contraction and release in the muscle cells. And we'll be looking at how this charge change results in a signal that we can actually see from entirely outside the cell. To study the action potential itself, you need to measure the voltage across the membrane, which basically means sticking an electrode into the cytosol. And that's not what we want to do when we're doing an EKG or something. We don't want to be sticking probes into individual cells in somebody's heart. It is how the mechanism here of the ion pumps and the ion channels was figured out, was from sticking probes into individual cells. There's a whole field of science known as electrophysiology, which basically studies the electrical behavior of cells, and a lot of that is based on the ion pumps and ion channels. A lot of that work, incidentally, was done with giant squid neurons, because the neurons of the giant squids are also giant neurons. They have large axons that you could actually stick fairly crude electrodes into and measure the potential across the cell membrane without needing really fussy work. I mean, if you tried to work with mammalian neurons, you can do that now, but it requires much, much finer control of getting tiny, tiny electrodes into the cells. So electrophysiology is the study of things like the action potential and how it's triggered and how the various channels work. This simplification I've given here is only really a tiny fraction of the whole story. There's entire courses on how neurons and muscle cells behave electrically. It's not, however, this signal, I mean, it's the cause for the signal that we see in electrocardiograms, but it isn't actually directly observable. And so we will talk in a subsequent video about the signals that we can actually see with the electrocardiogram and how they're derived from these action potentials.